So many of us know that the Pima Indians who live in southern Arizona have the world's highest incidence and prevalence of type 2 diabetes. Shown in the panel here is the prevalence of type 2 diabetes among adults living in the United States, and you can see that Pima Indians have a higher rate than other American Indian tribes such as Navajos, as well as Hispanics, blacks and whites in the United States. Pimas also tend to develop their diabetes with a younger age of onset. As you can see here in both males and females, that Pimas, by the time they're age 45, more than 50% of them will have already been diagnosed with type 2 diabetes. So because of this high rate of diabetes, the NIH set up a branch of NIDDK to specifically study diabetes among this American Indian tribe. Our early studies began on trailers in the Gila River Indian community shown here, and here's a historic photo from probably the late 60s of the staff working on the reservation. From 1965 to 2007, we conducted a longitudinal study of type 2 diabetes among members of the Gila River Indian community, and this is just south of Phoenix, where we asked all community members from the time they're age 5 and then every 2 years thereafter to come in to this outpatient clinic on the reservation for a medical exam. What was measured as part of this exam that's important for today's talk is height and weight, so we knew BMI of an individual at each visit. They would get a 2-hour oral glucose tolerance test, so we knew if they were diabetic and at what age they developed diabetes. We'd gain information on their families so we could put together pedigrees. We'd also draw blood for DNA analysis. And to date, we have about 7,800 individuals from this community that have DNA and longitudinal phenotypic data. Then in 1982, the NIH added an inpatient clinical research center to our branch in Phoenix, and this is located within the city of Phoenix, where we began doing studies on Pima Indians before they developed type 2 diabetes to try to understand what are the metabolic determinants that can later predict type 2 diabetes onset. So we have DNA and phenotypic data available on about 550 American Indians that have been categorized for various things, including their glucose and insulin responses to an oral glucose tolerance test, their percent body fat measured by DEXA, their acute insulin response to a 25-gram intravenous glucose bolus, insulin action measured by a hyperinsulinic euglycemic clamp, energy expenditure measured both using a human metabolic chamber as well as ventilated hood studies. And we also have tissue biopsies available on a subset of these individuals, again, before they develop diabetes. And we have subcutaneous adipose biopsies available on 247 volunteers and skeletal muscle on 248. And these have already been analyzed previously for gene expression using the Affymetrix exon arrays. So type 2 diabetes in Pima Indians, similar to what's seen in other ethnic groups, has a strong heritable component. What you see in the upper left hand is looking at individuals who all live in the Gila River Indian community. So all of these individuals are basically of the same socioeconomic class, same levels of income, lifestyle, and yet individuals on the reservation who are full Pima have a much higher rate than half Pima or non-Pima. So this suggests that there's a strong genetic component in this disease as well. So that's the role that I in my group play in the branch, is to try to identify this genetic basis for type 2 diabetes and its complications in Pimas. So most of our work has been focused on SNP associations, and I'll be talking about that work today, but we've also done some epigenetic studies, including genome-wide methylation studies. So we've previously done a genome-wide association study using the Affy 6.0 array, and these are our results when we looked at type 2 diabetes. We ultimately looked at about 7,500 individuals, and our top hit sat in this gene KCNQ1, which I'll come back to, and our second hit was sitting in this gene DINER, which we were able to do some functional studies and show that it plays an important role in the Notch pathway signaling, in the Notch signaling pathway, which is important for pancreatic development. And this variant was not associated with diabetes in Caucasians, but Caucasians had a different gene in the same pathway, Notch2, which was an established gene in Caucasians. So we seem to have two different genes, but converging on the same pathway. Recently, this exact variant has been replicated in a small case control study for type 2 diabetes among East Asians. We also used the Affy 6.0 array to look for variants that increase risk for BMI, and we looked at about 6,700 individuals. Our top hit was in the intergenic region, and our second hit was in the MAP2 kinase 3 gene. This gene is important for cytokines stimulating P38 to elicit immune and inflammatory responses. When we first found this variant, no other groups had reported it, but we contacted the GIANT consortium that has about 250,000 Caucasian samples at the time. They also were able to find an association, and when we did a meta-analysis between our American Indian data and the GIANT data, the joint p-value was about 10 to the minus 9 for BMI. So this was our top diabetes hit. It mapped to this gene, casein Q1, and this encodes an islet pore-forming alpha subunit voltage-gated potassium channel, so it should be involved with insulin secretory function. Variations in this gene were first identified in GWASs that came out of East Asians and were later shown to also have an effect on Caucasians. After this was identified as an important gene for type 2 diabetes, the DECODE study in Iceland, who also have family data, were able to show that there's a very strong parent of origin effect on the effect of SNPs in casein Q1, which sits in an imprinted region. And this is looking at parent of origin effects of our SNPs in Pima Indians, since we also have family data. So this particular variant is a C to T substitution. So you can see that individuals who inherit the C allele from their mother, either as a homozygote or as a heterozygote, where they got the C from mom, their association with type 2 diabetes approaches 10 to the minus 12, with an odds ratio of 1.92. If you're a heterozygote and got the C allele from the dad, your association with diabetes is completely non-significant, odds ratio 0.93. Again, this gene is predicted to affect insulin secretion, and we also have family data looking at insulin secretory function. So this is looking at data here among Pima Indians when they're still normal glucose tolerant, so they don't have diabetes yet. And this is looking at their insulin response to a 25-gram intravenous glucose bolus. And down here, you can see that those who inherited the C allele from their mother have markedly decreased insulin secretory function from 0 to 10 minutes compared to those who got the C allele from the dad. This single locus, if you take into account parent of origin, accounts for 4% of the variance and liability to type 2 diabetes in Pima. For those of you who don't study polygenic diseases, 4% might not seem like a lot, but it is. In Caucasians, you need about 100 genes to get maybe 2% of the variation accounted for. But we wondered what we were missing by using the Affy 6.0 array to do studies in our American Indians. This is looking at the population substructure of Pima Indians, comparing it to other HapMap populations. So in the upper panel, if we assume that these ethnic groups were derived from 3 populations shown here, we can look at Pima Indians, which are shown in the very last column, and we can see that Pima Indians, it looks like their genome is about 58% derived from East Asians, 41% Caucasians. But now if we assume that there's a 4 population substructure, you can see that Pima Indians have this huge purple chunk, which is completely unique from these other populations, and about 91% of the Pima's genome is this complete unique aspect, which is shared only with Mexicans, which is shown in the panel next to it. If you see 5 populations, it's pretty stable. So we're wondering, what are we missing by studying an array that isn't going to capture the American aspects of the genome? So we wanted to develop a platform to better interrogate the Pima genome. So our goal was to design an Axiom genotyping array to capture all the common variation, which I'm defining as greater than 5%. That's non-redundant, meaning that that variants share an R-squared of less than 0.85 that are within 500 kb windows that we detected in some of our whole genome sequence data. We'd previously attained whole genome sequence data at about 40x coverage from Illumina. And for this particular chip, we decided we wanted to focus only on variation that was detected in full heritage Pimas. So we analyzed genomes from 266 Pimas who were 8/8 Pima, meaning that their great-grandparents were full heritage Pima as well. So among these individuals, we detected about 9.7 million variants. Of that, 4.9 million had a frequency of greater than 5%, which we wanted to zoom in on. But we also didn't want to miss some potentially functional variation that fell a little bit below the 5% frequency. So we went in and captured variation that fell between 1 to 5%, but that was in a coding region and was predicted to either cause a missense frameshift or splice site variation. So we had about 66,000 more variants that we wanted to look at. So we had a total about 5 million variants that we wanted to target. So to do this, we could actually tag out about 4.4 million variants using markers that Affymetrix had previously had some experience with on these genotypic arrays, and they were known to be good performance markers. So our 4.4 million markers of variants could be tagged out using about 410,000 good performance markers that we could put on the array. We also captured an additional about 358,000 variants that could be captured by markers that hadn't been used by Affymetrix before, but based on their sequence they predicted to should separate well. And so these 358,000 SNPs could be tagged out using 294,000 markers. And because these markers had never been used before, we added some to add redundancy. So ultimately, we were able to tag out about 4.8 million SNPs using about 708,000 markers. And so we obtained, if everything worked, we would have gotten 96% coverage of what we wanted to look at in the Pima genome. The samples that we wanted to use with this Axiom array is basically everybody who we had DNA and phenotypic data from the Gila River Indian community. But we genotyped in 2 different stages. So our stage 1, we selected those individuals who were full heritage Pima, and we had about 3,700 of these individuals. 42% of them had type 2 diabetes. After we got the data back and looked at it, we decided, and got more money, we decided to move forward with a stage 2 analysis, which included all the other DNA samples that we had. And that was about 4,100 additional people from the Gila River community. These individuals are about half Pima and an additional 1/4 some other American tribes, so they are about 3/4 American Indian. Their rate of diabetes in the second sample is about 20%. So this is looking at the genotypic quality of the markers that were put on this chip. And basically the bottom line is about 77% of them passed and gave us clean quality genotypic data, which we can bring forward for association analysis. Our biggest chunk of failure up here was SNPs that just didn't cluster well and couldn't be called. And this, I think, is where we're using a lot of markers with performance unknown that didn't have optimal design scores. Our second biggest chunk of failure was 4% of the SNPs came back as being monomorphic that we had called as being polymorphic in our whole genome sequence data. When we went to look back at our whole genome sequence data, a lot of these SNPs were sitting in repetitive elements. So indeed, we're not sure if they were real SNPs or not, so they may represent some sequencing error. This is comparing the SNPs that gave good quality genotypic data, looking at the allele frequency, comparing Pimas to Caucasians. So this green chunk shows SNPs that were more common, meaning their minor allele frequency was more common in Pimas compared to Caucasians. The chunk here are SNPs that were more common in Caucasians than Pimas. This chunk represents SNPs that the allele frequencies were within 5% of each other between Pimas and Caucasians. But I think the most interesting categories are these dark and light lavender, because these represent SNPs that are completely monomorphic in Caucasians. The larger chunk are SNPs that had a frequency of greater than 5% in our American Indians, and the lighter one were SNPs that had a frequency of less than 5% in our American Indians. But again, all these were monomorphic in Caucasians. We next wanted to know, among the SNPs that passed, how well did they capture everything that we wanted to look at in our whole genome sequence data. So this is comparing our custom array to the Affy 6.0 array. So if we look at variations that had a frequency greater than 5% from our whole genome sequence data, we're actually able to capture 91% of those using our custom array, whereas the 6.0 array captured about 77% of our variants. Where we really saw a difference was in our SNPs that had a frequency between 1 to 5%. And again, most of these represent coding SNPs, that we were able to capture 56% of SNPs in this range using our custom array, and only 24% occurred on the Axiom, or in the commercially available 6.0 array. So we're starting to use our custom array data to do some genome-wide association analysis, and we're still starting to plow through this. Our stage 2 data just came back in July. But some of the things that we've done is we've looked at our top SNPs to see those that are associated with type 2 diabetes, and I'll mention some of these today. I also have a poster on Friday on these. We've also looked to see which SNPs associate with BMI, and we looked at BMI both as in adulthood as well as childhood BMI. We've looked at metabolic determinants of type 2 diabetes, namely insulin secretion, insulin action, and percent body fat. We also have looked at metabolic determinants of BMI, namely energy expenditure, and Paolo Piaggi, who's a fellow in my group, has a poster on Thursday where he's identified a variation that results in a decrease in energy expenditure, and people who carry this variant have increased BMI. as a consequence of their decreased energy expenditure. And the risk allele frequency for this SNP is 60% in Pimas and completely monomorphic in Caucasians that don't carry it. And we've also looked at a GWAS for diabetic nephropathy, and Avica Matsadova in my lab has a poster on Friday describing some of these results. In addition to just looking at our strongest p-values, we wanted to directly look at some of our coding variation, which was a little rarer, but that sat in really good candidate genes that might not have the statistical power to give our really robust p-values. So this is looking at our top hits for our GWAS for type 2 diabetes. So the SNPs that were identified, their position, the locus, their p-value after adjusting for age, sex, birth year, family membership, and Indian ancestry, the p-value after applying a genomic control, the odds ratio, and then I wanted to show that each of these variants were also independently associated with diabetes in our stage 1 and our stage 2, so we have independent replication. You can see that our top 2 hits sit in this gene casein Q1, which we already knew was a really big gene in Pima Indians. The SNP that is shown at the bottom is a SNP that we were already aware of from our previous studies, but we were always a little baffled why this gene had such a strong effect in our American Indian samples, but not so strong in other ethnic groups when the allele frequencies were all quite comparable for the risk allele, which is shown here in red. This top SNP ends up being our top GWAS hit using the custom array, and this SNP is brand new to us. We were not aware of this variant, and we're really excited to see that the risk allele frequency for this variant has a frequency of 40% in Pima Indians, 33% in the other individuals from our Gila River, and yet is very rare in other ethnic groups until you get to Mexicans. We haven't looked at parent of origin effects for this SNP yet, but I expect that we will get some very robust associations when we do that. So this is exactly what we were hoping to find with this array. Another 2 SNPs that we found in our top 7 both map to this gene TH. And this we found that the genotypes had an effect with a gender interaction for these particular SNPs. So let's look more closely at them. So TH encodes the tyrosine hydroxylase, which is a rate-limiting step in the synthesis of catecholamines. So this is looking at one of those 2 SNPs broken down into females and males, where C, shown in red, is the risk allele, so the homozygous, heterozygous, homozygous for the non-risk allele. And you can see that among females, that individuals who are GG have an increased prevalence of type 2 diabetes across all age groups compared to heterozygotes or individuals that don't carry the risk allele. The p-value is approaching 10 to the minus 9, odds ratio 1.29. In males, we see no association whatsoever with this variant. We looked at this SNP. It's not associated with percent body fat. It's not associated with insulin sensitivity. But let's look now at the insulin secretory function based on this SNP breaking down into males and females. So these upper 4 panels are looking at insulins and glucoses during an oral 2-hour oral glucose tolerance test. And you can see here that the 30-minute insulin, so early insulin secretion, is markedly reduced in the females that are carrying the risk allele for eventually developing type 2 diabetes, and these individuals are still normal glucose tolerant who are being typed here. So they have a significantly lower early insulin secretion, resulting in a significantly higher 60-minute glucose, which is what you'd expect. We see this in females, not males. If we now look at the intravenous glucose tolerance test, again among normal glucose tolerance Pima Indians, you can see among females that impaired insulin secretion all the way from 0 to 10 minutes, not seen in males. This SNP or these 2 SNPs that we found are actually relatively close to casein Q1, where they sit about a megabase away from casein Q1 on chromosome 11p. So here's our top signal, and here's our second top signal sitting here, but we've been able to show that these are independent signals. We're not just catching the same thing. Casein Q1 is, I think, a well-established type 2 diabetes locus. TH, I don't think, is widely recognized as being a type 2 diabetes locus yet, but when I did look in the literature, I found that in 2012 there was a report in American Journal of Human Genetics where they did a very large study in Caucasians where they looked at gene-centric SNPs, and they did a meta-analysis of 39 studies, which included 22,000 cases and 95,000 controls for type 2 diabetes. And they did identify a p-value of 10 to the minus 6, which met study-wide significance for them. So I think where our data is able to show that indeed this does seem to be an established type 2 diabetes locus here, and in our data at least, it shows a very strong paired of origin effect. Let me turn now into analysis of some of the rarer SNPs that were on the chip, but because they were rare, they might not reach genome-wide significance. So the two genes that I'm going to mention today are ABCC8 and KCNJ11, which encode the SIR1 and KIR6.2 subunits of the beta cell potassium channel. Rare missense mutations in these genes have been shown to cause rare forms of monogenic permanent neonatal diabetes, as well as hyperinsulinemia hypoglycemia of infancy, which is also called HHI. And a common variation in E to K in KCNJ11 is an established type 2 diabetes variant for polygenic diabetes in many ethnic groups, but not Pima Indians. So these are the subgenes that encode the subunits here of this potassium channel, where under low glucose conditions, this channel is open, potassium flows out of the cell, you get hyperpolarization, your calcium channel is closed, and you don't get insulin secretion. Under high levels of glucose, this potassium channel is closed, you get depolarization, calcium flows in, and you get insulin secretion. So this is the subunit of the channel we're talking about. So this is the missense variants that we identified in these two genes. We have 9 of them here, 2 in KCNJ11, 7 in ABCC8. Whether or not the variant is novel, and 6 out of the 9 were novel into our study. The amino acid that's predicted, the frequency in our American Indian sample, the odds ratio for association with diabetes in 7,700 individuals, and the p-value. And you can see that the SNP that jumps out is this arginine to histidine, shown here, that has an odds ratio of 2.02, p-value 10 to the minus 5. This variant it's novel, and we were able to do some functional studies and show that it's inactivating in vitro, and it's carried by 3.3% of the individuals living in the Gila River Indian community. So this is looking at the diabetes prevalence. So prevalence shown here by different age groups of people, and you can see by the purple line that individuals who are heterozygous carriers for this variation have an increased risk for type 2 diabetes across all ages. These individuals also develop type 2 diabetes when they're leaner. So here is looking at the cumulative incidence of diabetes by the maximum BMI, and again follow the purple line, and they are developing their diabetes at a leaner BMI. We are also able to show that these individuals had a meaner, younger age of onset by 7 years compared to people who developed diabetes without this variant. Because rare mutations in this gene have been associated with hyperinsulinemia, hypoglycemia of infancy, we were wondering if these variations were also affecting insulin secretory function when the baby was still in utero. Andrew Hattersley has a fetal insulin hypothesis that says high fetal insulin results in higher birth weight, and this has been documented in several studies. So we use birth weight as an indicator for whether or not these kids were hypersecreting insulin in utero. This is looking at the association data for all the SNPs that we detected with birth weight. And again, this arginine to histidine is the one that jumps out as being associated with increased birth weight. So we suspect that these babies are over-secreting insulin even in utero. With a carrier rate, 1 on 30 in Pima Indians, that would predict that 1 out of 3,600 births would be homozygous for this variant. And indeed, we were able to identify one individual who was a homozygous carrier. This is looking at the pedigree of this person here, and here's the carrier here. When we went back and retrospectively looked at this person's medical charts, we found that indeed he had been diagnosed with hyperinsulinemia, hypoglycemia of infancy at 4 months of age, had very severe seizures as a result, and developed type 2 diabetes by the age of 3. So he had a very, very severe phenotype in homozygosity. So to summarize, our custom-designed genotyping array that was entirely designed based on whole genome sequence data from Pima Indians has identified a new signal in the established type 2 diabetes gene casein Q1 that's much more common in American Indians and Mexicans as compared to other ethnic groups. We've confirmed that TH is a valid type 2 diabetes locus. In our American Indians, we've shown that there's a strong gender effect on type 2 diabetes and insulin secretion. And lastly, we've identified a Pima Indian-specific inactivating missense variant in ABCC8, where heterozygous carriers have a doubled risk for type 2 diabetes with a 7-year younger age of onset, and homozygosity is predicted to have a very severe phenotype of HHI. So I'd like to acknowledge the members of the Gila River Indian community who have volunteered for decades, and much of what is known, especially clinically, about type 2 diabetes has come from some of the early studies in Pima Indians. I would also like to acknowledge all members of our branch in Phoenix, because everybody at the branch has had some hand in the research that's been presented today, as well as thank Affymetrix, who helped design this chip for us. And this work was supported by the intramural program of NIDDK. Thanks.